All right, everybody, welcome to AP Biology. This is our final video on cell reproduction. And specifically, we're looking at sexual reproduction and meiosis with this video. So remember, mitosis creates two identical daughter cells, while meiosis creates four genetically different daughter cells with half of the genetic information in them. And binary fission is an example of asexual reproduction. And meiosis is a concept involved in sexual reproduction, where we're creating those sperm in the egg, we're creating genetic variation. Now, if you guys look at the video here on the chromosomes, I want to make sure that we understand the difference or the special component of meiosis, which is the chromosomes are matched in these homologous pairs, and specifically all somatic or body cells. So every cell in the body that's not sperm or egg is considered a somatic cell. They have a specific number of chromosomes inside them. So for example, our human cells have 46 chromosomes inside them. 23 are from mom, 23 are from your dad, and they're matched up together in 23 sets of homologous pairs. And that's inside of all of your cells that are not sperm or egg, and those are known as somatic cells. And so let's look at this on the next slide. So here is our chromosomes. Here's our homologous pairs. And we say that the homologous chromosomes have the same genes for the same characteristics and the same traits on the same place at the same location, which is known as a locus or loci. You're going to notice in this diagram that one copy of the chromosome is from the mom and one copy is from the dad, and they're duplicated because they've been through the S phase of interphase in the cell cycle. And so here, this is like a condensed or cartoon version of how DNA is wrapped around those histone proteins as a result of the S phase. And so we can see these duplicated chromosomes that we now call sister chromatids matched up to each other as homologous pairs. So when we talk about gametes, which are sperm and egg, we have to remember they only have one set of DNA. Or half of our chromosomes. And so we have to remember that all somatic cells have two sets of chromosomes, and we call them diploid or di, which means two. Gametes, on the other hand, are haploid or have half the DNA. And so they only have one set of chromosomes. And let's look at this on the next slide. So here is our diagram of kind of our sexual life cycle, if you will. And you'll notice that we start off as, let's look up at our gametes, our egg and our sperm at the very top, like 12 o'clock. And they undergo fertilization where they combine and they put together their 23 chromosomes from each to make one zygote or diploid cell with 46 chromosomes in it. That zygote reproduces through mitosis until eventually we get to the place where we're ready to sexually reproduce. We create our sperm and our egg through the process of meiosis, and we cut the amount of DNA in half so that they are haploid or have 23 copies. Now, meiosis reduces the number of chromosomes from diploid to haploid. And so meiosis, just like mitosis, is preceded by chromosome duplication of the S phase in interphase. But in meiosis, we're going to undergo two sets of cell division. So we get four daughter cells and not two. So if we were to look at a graphical kind of representation of how mitosis and meiosis are different, you can see here that we're starting with our duplicated chromosomes. Specifically in meiosis, we're going to have our crossing over. So again, notice the difference between mitosis ends with two cells, meiosis ends with four. Mitosis, all of the chromosomes line up in the middle, and the sister chromatids are what are being separated in anaphase, whereas the sister chromatids don't actually separate until anaphase 2, the second set of divisions. And while they organize themselves as homologous pairs in the original step of meiosis. So when we go through this in detail, I want you guys to know the first division is meiosis 1. And basically we start with synapsis, which is the pairing of homologous chromosomes. And then they're going to cross over. They're going to exchange little bits of themselves, as you can see here. So here we have the father's DNA, the mother's DNA, your chromosome that you got from your dad, the chromosome you got from your mom. And they hang out together as a homologous pair. They get so close that they actually swap DNA. And so what happens when you look at it after they're done, the dad DNA now has one of the original DNA strands, but the other one has a little bit of mom on it. And the same thing has happened over here on the maternal side. So we've exchanged genetic material. And that's going to be happening in meiosis 1. We're going to separate the homologous pairs to make our two daughter cells each have a complete set of chromosomes. And then meiosis 2 is the one that looks almost identical to mitosis, where we're actually physically separating those individual sister chromatids so that we end up with 4 unique haploid cells, half the amount of DNA. So here it is in kind of a picture format. You can see here that homologous chromosomes are going to be separating after crossing over occurs, and we're going to end up with 4 different bands of DNA that now exist. Then what happens is there's no more interphase. After we go through meiosis 1, we immediately go directly into meiosis 2. And in doing so, we'll separate those sister chromatids and we'll end up with our 4 haploid cells, half the amount of DNA. Now, again, we can compare mitosis and meiosis using this chart. But what we're going to see is that the differences are more in the homologous pairs, the crossing over, and the fact that not having 1 division, but rather we have 2 divisions in meiosis. So what happens is that we create 4 unique cells, and those 4 unique cells are the result of what's called independent orientation of the chromosomes. And so it allows for this random fertilization to occur because any one of the 4 eggs we create can be fertilized by any one of the 4 sperm that get produced every time, each time creating a unique combination. The other thing that's happening is when we have homologous chromosomes, they line up with each other, but the maternal doesn't just line up on left and the paternal on right. They can be in different directions. And so it allows for random assortment of DNA as a result of that crossing over. So let's look at it. Here's our random assortment of chromosomes. Notice, let's say the blue is paternal and the red is maternal. Notice that we have the blue and the red, and we allow all of this different combinations to occur in our sperm and egg or in our gametes that are produced. Random fertilization by the sperm and the egg then add to that diversity even more, okay? The other thing that I want us to make sure we understand about those homologous chromosomes as they kind of match up is that they actually, while they have the same genes in the same location, they don't have the exact same alleles. So we have to go back to our understanding of genes and alleles. On each gene is a chromosome, or each chromosome is a gene, that we have 2 different alleles, and they can be different. So we can actually inherit a dominant allele, a recessive allele. We can have 2 of the same, we can have 1 of each, or maybe we have 2 recessive. But again, when we have that crossing over and then we have that random assortment of how they line up, we're able to create a lot of genetic combinations inside our sperm and egg as a result of our meiosis. And so we can talk about how crossing over creates even more genetic variation, which is good for survival and evolution. And again, that crossing over is during meiosis of prophase 1 of meiosis 1. And because again, we're not just separating where mom and dad line up, we're actually exchanging genetic material between mom and dad. So here's an example. If we look at 8.18, we see that mom has the dominant C and E alleles, while dad has the recessive C and E alleles. Now, due to the crossing over between mom and dad's DNA, the homologous pairs, what happens is we end up creating a sister chromatid that has a C and an E dominant alleles, just like it was originally in the mom. We end up creating a dominant C with a lowercase e, which is a combination of the mom and dad on 1 sister chromatid. We end up creating another CE combination that is a combination between mom and dad as a result of crossing over. And then we have the 2 recessive alleles as a result of dad's original paternal type of chromosome or sister chromatid. So we separate, we create that genetic diversity as a result of crossing over. Now, if we want to talk kind of big picture about our genetics, we can talk about or use a karyotype. And a karyotype is a photographic inventory of an individual's chromosomes. And it actually organizes us by our chromosomes and how they match up. And the only 2 weirdos is our X chromosome and our Y chromosome, because they don't look alike and they don't carry the same genes, but they're the only 2 kind of loners at the party. So they hang out together. So how do we get a karyotype? Well, we take out some blood and we isolate the DNA and we open it all up and we sort it, and then we sort it by size and shape. And you'll notice that they perfectly match up in homologous pairs, except that X and Y, which are the little funky bunch at the end that hang out. However, using a karyotype, we can identify some genetic disorders. For example, if we have a whole extra copy of chromosome 21 because it didn't separate in anaphase, we can have what's called trisomy 21, which is Down syndrome. And that can be a type of disorder as a result of what's called nondisjunction. So here's trisomy 21. Notice they have 3 copies instead of the 2 that you're supposed to have. The chance of having a child with Down syndrome actually increases with mom's age. And so anybody over 35 years of age is considered a geriatric pregnancy. And that just means we have to be a little more careful, and there's an increased risk of trisomy 21. And so again, trisomy 21 is a result of an accident or a mistake during meiosis. And so sometimes when the homologous pairs or the sister chromatids don't separate, it's called nondisjunction. And now these 2 that didn't separate get inherited together, giving us an entirely extra chromosome. So here you can see our nondisjunction, our homologous pair, they stay together and we end up with an extra set of DNA. Now, the other thing that can happen is we then have that egg with an extra chromosome and it gets fertilized by the sperm. And now we have a zygote or our baby with an extra chromosome. We have our birth defects. There are other disorders that come from having different numbers of sex chromosomes. Nondisjunction can actually produce gametes that have an extra X, an extra Y, or maybe they're even missing one of their sex chromosomes. And so there can be results of that, like poor ability to grow a beard, or maybe they develop breasts instead of underdeveloped or regularly developed testicles. All of those things can happen as a result of trisomy or nondisjunction. And so here's some of the examples of human sex chromosomes that happen as a result of nondisjunction: Klinefelter's, and Turner syndrome. But you'll notice that sometimes when you have trisomy or an extra set of chromosomes, it has no effect overall. All right, you guys. Alterations in chromosome can also cause birth defects or cancer, as we've previously studied. We can have deletions where we actually delete part of our DNA. We can have duplications. We can invert where we actually switch the location of genes. And we can have translocations where an entire set of DNA hops onto a different chromosome. And that's a problem. All right, last one. That's it. The bell rang. We're all done. I'll see you guys back in class.